Okay, students, so we are continuing on the where we left off in the last class. That is, we are talking about enzymes that will digesting enzymes that will cut off peptide chains. So, because it has been a while, I'll just repeat what we are thinking about here. So, we are talking about protein sequencing. So, protein sequencing is the idea of let's say we have a long protein chain, and we want to identify what are the individual amino acids that are involved here and in what order, right? So they say this is composed of lysine, arginine, then methionine, then glycine, and so on. So we want to know what is the order and what are the specific amino acids that are present here. So, and we already know that the N terminal is on this side and C terminal is on this side. Okay. So what I initially talked about is that the two steps of doing this is, the first step is we usually find out, so this is n number of amino acids are here. First thing we do, we find out what is the N terminal amino acids. Okay, so using the chemical method, and that leaves us the rest. So this is arginine, methionine, glycine, and so on. So now the number of amino acids here are n minus 1. The next step, what we do, we also treat this with the DTT or BME, which is a thiol. So what happens here, if there is a cysteine here, so cysteines can form dipeptide linkages. So those are broken off using this process. Okay, so that is cysteine breaking off. So they become SH and SH. Okay, so that is that. Okay, so next, what we do, we treat this with this enzymes, this proteases, which then breaks up this long chain into smaller chains. So these are some of the enzymes. So we break this up into smaller peptides, and then we identify the smaller peptides using mass spec, and then tell that what are the arrangement of the peptides that we'll come to in a moment. So these enzymes, what do they work? So these are basically what this is. This is a cleavage point. Okay, so trypsin, for example, I'll give an example. So let's say this is an amino acid chain, a peptide chain, which looks like this, which is let's say methionine, histidine, okay, and followed by glycine, followed by lysine, followed by leucine, followed by isoleucine, followed by arginine, followed by glutamic acid, followed by let's say what else we can put here. Let's say we followed by cysteine, and so on. Okay, so this is the C terminal amino acid. So this is the last one. So once you do the trypsin digest on this, this is how it's going to look like. So it is going to create or cut wherever there is a lysine or arginine present. Okay, so this is how it's going to look like. So it's going to cut here. Okay, it's going to cut here, and then it is producing the peptides where the lysine or arginine is present in the C terminus. Okay, so the product is going to look like this. So methionine, histidine, glycine, lysine, right? So lysine being the C terminus. It's going to produce leucine, isoleucine, arginine. It's going to produce glutamic acid, and cysteine is the third product. Okay, so that is the product that will be produced. Okay, now this method alone does not really tell you that if this was the first or this was the first. So you cannot really go back and reassemble this as the original peptide. So we have to do at least two digesting methods. So we usually do two enzymes, or one chemical and one biological digestion, to get the complete picture. So this alone does not tell you. So this only tells you that this fragment being the C terminal fragment, because there are no arginine or cysteine, lysine here. So this must be the C terminal fragment. Between these two, any of them can be the N terminal fragment. That we cannot tell from this method. But this is basically how these proteases, they digest and create smaller fragments. Okay. Now let's look at a problem where people have done this method, and they have fragmented and found out and then reassembled the original peptide. Okay, So this is a original polypeptide. Okay, so first thing, so this is the problem what they are doing. So first thing, they are simply hydrolyzing. So this is done using concentrated HCl. So what it tells you is that there are this many amino acids of alanine. Okay, and let's say this is the breakdown of all amino acids and how many numbers are present. Okay, but this does not really tell you anything regarding the order. Okay, so this just is the total number. Then what it did, this same pep protein, they react with FD and B. So remember this, what it does, this reacts with the N-terminal amino acid. So the product is, if you identify, this is 2-nitro diphenyl, so 2,4-dinitrophenyl glutamate. So that means that the glutamic acid is the N-terminal amino acid. Okay, so that is that. So glutamic acid being the N-terminal amino acid. Okay, so that is the only thing that we have known so far. Then what it does the reduce the disulfide bond. This does not really do specifically to anything to identify the protein, but it is important for the next steps. Okay, so then after the disulfide bond is cleaved, it is treated with a trypsin. So trypsin now generates this fragment. So you can see here that K stands for the lysine and R stands for the arginine, right? So all fragments except one has lysine or arginine at the C terminal. So lysine and arginine, lysine, but this fragment does not have any C terminal lysine or arginine, which means that this must be part of the C terminal. Okay, so the peptide chain must be ending with this sequence. The second thing what it did, then also, as I said, they need to do another method, which is another digestion method. So in this case, they use the cyanogen bromide. So cyanogen bromide produces methionines at the C terminal. So two fragments are produced. One at the both of them had the methionine at the C terminal. Another fragment which does not have any methionine at the C terminal. So that must be the final C terminal peptide, part of the peptide. Okay, so you can see that there are considerable overlap in terms of the sequence between these two. So I'm talking about these two. So you can see this is D, S, H, then V, then C and D. So up until this point, this is identical for both of them. So this must be the C terminal peptide. So I'm just writing this down. So D, C, V, H, S, D. Okay, so this is the C terminal peptide. Now what we have here is that, so now we have to find out what are the overlapping sequence between this and this and this fragments. Okay, and the basis of that we will figure out what is the final sequence. Okay, so if you see here is that the C3, which is right here, okay, so C3, that has a sequence overlap with the T1 and the T4. Okay, let me show you how that is. So if you look at this part, YLIACGPM, right? So this is right here, YLIACGPM, and then followed by two more. This is also similar with here. Okay, so that is, you can see here that this part of the sequence is right here, that is ALIK. Okay, so that is that ALIK part. Okay, so that is that. So what we can do, we can arrange this in a continuous chain. So we'll find out what is the ultimate thing, but let's see. So this, let's start with the T1 fragment, GAS, okay, MALIK, GAS MALIK. Okay, so that is the T1 fragment, right? So now followed by the ALIK. Okay, so you have then this fragment. right? So ALIK ends here. Then you have the YLY. So I'm going to use a different color right here. Okay, maybe not. I'll continue use the black, but I will use a different marker later. Okay, so this is the T1, right? Then you have the YLIA, YLIA, CGPM, CGPM. Okay, so YLIA, CGPM. Okay, so when the CGPM ends, then you have the TK, right? So then you have the TK from this fragment. So now let's identify the fragments one at a time. Okay, so I'm going to use a different color right here. So this part starting from ALIK to CGPM, ALIK to CGPM is the C3 fragment, right? Then followed by, if I use the green, so starting from YLIA and then TK, YLIA and then TK, this part is the T4 fragment. Okay, so now what do we have after that? So after that, let's see, then you can find out that the T2 and the C1 has some overlapping sequences, right? So both of them starts with this EGAA and ends with the DPR. So after that, the DPR, you have the GASM. Okay, so we can identify that sequence together. So let me go back to the black color again, and we'll see what comes out of that. So E G A Y H D. So I'm writing the T2 sequence. E G A Y H D F E P I D P R. Okay, DPR, and then followed by GASM. Okay, so this part is the T2, and this whole thing is then part of the C1. Okay, this whole thing is part of the C1 sequence. Okay, so that is that. Now what we have to do, we have to find out if there is any common sequence between them. Okay, or then we can find out the rest of the fragments, or anything with this fragment also. Okay, now try to see that if we can find any common sequence. So if you look at this two sequence carefully, see if there is any common one. So let's see carefully. So so what you have to figure out, which of them goes to the N terminal and which of them goes to the C terminal. Okay, so which of them comes first and which of them comes second. So how do you decide that? So what we have to do, you can see this GASM sequence is part of the GASM sequence right here. Okay, so you can see here that the GASM sequence is the part of the GASM sequence right here. Okay, so let me take out this color. So if we now put them in the order, so GASM is this GASM sequence right here. If we put them in the order, so this is how it's going to look like. So E G A Y H D F E P I D, okay, P R G A S M, P R G A S M. So if then comes this part, G A S M, A L I K, Y L I A, C G P M T K. Okay, this is followed by the last part of the sequence, that is, you can see this is TK and this is the TK here, D, okay, so I'll write it separately, TK, DCV, DCV, HSD, okay, so that is the 3 fragments, okay. So fragment 1, if we say this is 2, this is 3, this is fragment number 1, this is 2, and this is 3, okay. So that is your entire peptide starting from here, E, then continues till here, TKD, and then continues here, okay. So that is that. So this is the answer. So just what I do, this looks like it is matching over here. So this is the fragment, you can see that is the entire sequence, and this is how you build up the complete peptides, okay. So that is the method how you do it, okay. So now, having done with the synthesis of the peptide, so what we must know is that proteins, we know that they can be synthesized, they are synthesized typically biologically. So we can also synthesize proteins using chemical methods, okay. So this is the person who first came up with this idea. So the idea how it is done, it is called a solid state peptide synthesis. The term that is typically used is called solid state peptide synthesis, okay, SPPS. Okay, so we can see here, this is the what the solid phase means. So this part is basically that solid phase, that part, okay. What is it, solid phase? Because it is an insoluble polystyrene bead, meaning this is not soluble in the solution where the reaction is taking place. So this is actually like a little plastic bead. On top of that, what you do, you put up this first group, okay. So this, you can see, this is a benzene ring, and then CH2, and then Cl. Then what you do, you first bring up your first amino acid. So where this doesn't look like any amino acid, but if you look carefully, this is the alpha amino group, where it is protected with a group called FMOC. Okay, I'll tell you in a minute why. This is the alpha carbon, this is CH, and the side chain here, and this is the carboxyl. Okay, the carboxyl's O minus reacts with this carbon and throws off the chlorine. So now your entire this part, first amino acid, is bind to this, okay. And the reason for putting the FMOC is, if you don't put the FMOC, there will be a free NH2. That NH2 can also react with the CH2, okay. So we don't want that, okay. So that is the reason. We put this group, FMOC group here. So this is called a protecting group, okay. So this FMOC group is called a protecting group. Okay, so protecting group. So you can see this term mentioned here, protected by FMOC group. Okay, so that is that. So next, what we do, this is your first peptide, the amino acid that has been added. So next, what we do, we add this chemical entity called dicyclohexyl carbodiimide. So this reagent, I mean, I'm not going to too much chemistry for this. What it does, this binds to this NH, reacts with this NH2 group, okay. And what it calls is that it increases the reactivity of this NH2 group, and as a result, when the next cycle, it increases the reactivity of the NH2 group. What happens when we add the next FMOC protected amino acid, this DCC reacts with the carboxyl. Okay, you can see this part is the carboxyl. Again, this is the alpha carbon, alpha amino, alpha carboxyl. So this O is now, it reacts with the GCC. So this, what happens is that this carbon's positive charge gets increased, or in other words, its electrophilicity increases. Okay, electrophilicity increases. So what it does, it tempts this NH to react with this carbon and throws off this DCC modified part. Okay, so you have now added 2. So this is your first NH that did this reaction. This is your second amino acid carboxyl that just participated in the reaction. So you can see the color wise it is distinguished. So this is the purple one and this is the yellow one. So that way then there comes the third one which reacts with the second DCC modified carboxyl. Okay, so that is the second, that is the next amino acid being added. Okay, so that is how the chain keeps on growing. Okay, so this is the chemical method for protein synthesis. So I will stop right here and we'll start from the protein folding approach in the next lecture. So that's all for today.